This is the fourth main video on the topic of evolution for the life sciences syllabus. And in this video, we're going to look at the formation of new species, which we call speciation. So we looked at the term species in a bit more detail during the population studies video. But when we want to define a species, we define a species as a group of organisms of a common ancestry that closely resemble each other and that can interbreed and produce viable offspring. So interbreed means they can breed among each other, and in doing so, in mating and in breeding, they produce viable offspring. Viable as in fertile, as in the offspring not only is likely to live to sexual maturity, but when it does so, it will be able to mate with another organism from that same species and reproduce. The evolutionary process during which new species form, as we said, is called speciation. And we can define speciation in more detail as the formation of species, which increases the range of organisms on Earth. And in doing so, it increases biodiversity. So the various species and how diverse species are on Earth. If we then compare extinction to speciation, extinction is the loss of species, and this decreases the range of organisms on Earth, and therefore it decreases biodiversity. So extinction and speciation are basically opposites. What is a population? A population is a group of individuals of the same species which occupy a particular habitat, and because they occupy the same habitat, they are able to crossbreed successfully. So if members of a population can crossbreed successfully, they're considered part of the same species. So usually species is a lot more, a broader term almost. Within a population, within a species, you'll have multiple populations. For example, lions, most African lions are all of the same species, but the Kruger population is a completely separate population to the lions in Zambia. The lions in Zambia and the lions in Kruger are not able to crossbreed because obviously there's a lot of cities, borders, etc. between them, which means they're not part of the same population. However, they are part of the same species because if you were to put them together and you were to make them breed, they would produce fertile offspring. So members of a species share a gene pool, and there's free gene flow between the populations of the same species. A population, as we said, is a group of organisms of the same species that live in a particular place at a particular time with the ability to freely interbreed. So if they asked you for the definition of a population, this is what you'd give. There is also a definition in the population studies video, which is quite similar and covers the same main concepts. When we look at a gene pool is the total number of alleles of all reproductive individuals in a particular population. So going back to our lion example, the gene pool of the Kruger lions is not the same gene pool as the gene pool of the Zambian lions, because the gene pool only occurs for a particular population. When we talk about gene flow, this is the exchange of alleles between populations. And obviously when gene flow occurs, between different populations, it will increase the gene pool of each population because there are more different alleles coming in. So it increases the diversity. So when we look at the formation of a new species, when a new species arises from an existing species, so from the ancestral species, these two species can no longer interbreed because they're two separate species. We have the ancestral species and we have this new species. There's no gene flow between them, and they'll become isolated from each other in terms of reproduction. So the term reproductive isolation, we're going to use it a lot. It's quite an important term, and it's very important to be able to understand it and to use it when you're explaining something like speciation. For reproductive isolation, obviously it's pretty self-explanatory. It is basically just that one species can no longer reproduce with another species. So this new species can no longer reproduce with this ancestral species. Reproductive isolation can occur when two species are separated geographically, so separated by something like water, land, mountains, etc., or even if they occupy the same area. And we're going to look at those two different types of speciation and how they both result in reproductive isolation. So the type of isolation that develops between the new species and the ancestral species preventing interbreeding is what we were calling reproductive isolation. So we can see here that this would be an example of the reproductive isolation when the species are separated geographically. We know that this here is a geographic barrier. So we just start with the original population, all in green. They're then separated by a geographic barrier. Over time, it's important to know that this takes a long time. It's not going to happen over one generation. When these two species are separated, there's no gene flow between the populations. This green population now has their gene pool of alleles, and this blue population has a different gene pool of alleles. They're not exchanging alleles across this geographic barrier, which means that when a mutation occurs or if natural selection happens, it may only affect the green population. If a mutation occurs here, it won't be transferred to the blue population. And over time, as these mutations occur and as these populations become more and more different, they eventually result in new species. Now, even if the geographic barrier was removed and these populations were allowed to mix, they wouldn't be able to interbreed because they're completely separate species. And it's important to know that just because there is a new species that has arisen from this ancestral species, it doesn't necessarily mean that the ancestral species has become extinct. It may be that only the blue population is left, but it could be that both populations are still remaining. So the formation of a new species occurs over a specific pattern. We have variation within a population. We then obviously have evolution by natural selection, which we spoke about in the previous video. These species then adapt to their environment. Reproductive isolation prevents interbreeding between these two or more populations, each of which has adapted differently to their environments. And because of this, we now have new species. So the variation within species, which we spoke about here, this is like the starting point for speciation. You can't have a speciation if all the animals in the species are exactly the same. And we know that, we spoke about that earlier. You need variation within a population for natural selection to occur. And you need natural selection to occur for species to evolve. So genetic variation occurs within a species with the result that organisms of the same species differ in appearance. Genetic variation, as I said, is essential for natural selection because the environment selects the best adapted organisms for survival from a wide range, sorry, the word range is missing there, of genetically different individuals. So some individuals are well adapted, some are not so well adapted. The two ways that genetic variation is mostly caused by is genetic recombination and mutations. Both of these we spoke about in previous videos, genetic recombination in the meiosis video, and mutations, I seem to remember that was mostly in the, it was actually in fact in the meiosis video and in the RNA video. So if we just have a quick recap, genetic recombination occurs during meiosis and during sexual reproduction. So during meiosis, when the gametes, the homologous chromosomes, cross over, forming bivalents, we have some genetic recombination here. When they are separated into new daughter cells, we also have more genetic variation, more genetic recombination, because we have various arrangements of the homologous chromosomes, which we know, even though it's depicted here as just red and blue, we know that they've already undergone crossing over. So they actually wouldn't just be, purely maternal and purely paternal. They'd have a little bit of maybe maternal over there, paternal up there, where crossing over occurred. And we can see that all the arrangements for the cells differ. So you might have all the maternal cells on one side, all paternal on the other. You might have chromosome 1 being maternal and the rest paternal, etc. And there's obviously a wide, an enormous range of combinations, like 2 to the power of 23 or something, which obviously is a massive number. That alone causes huge genetic recombination. Not even thinking about the more recombination that comes from crossing over. So when we just look at crossing over and at splitting into daughter cells, the term independent assortment can be used. We can also use random assortment, random segregation. It doesn't really matter what you use as long as you pick your term and stick to it. I mostly used independent assortment and random segregation as the two terms. Genetic recombination also occurs on top of crossing over and on top of splitting into daughter cells. It also occurs during reproduction because the sperm cell and the egg cell, there's obviously however many different egg cells and however many different sperm cells, and the number of combinations of those two fusing and combining is also extraordinary, a huge amount. So we can see that there's a lot of genetic recombination occurring. And this is obviously why the offspring of the same parents differ from each other, because they all have a unique gene combination. They all have a unique arrangement here, crossing over, unique sperm and egg cell that have combined. So we can see that there's a lot of variation just from genetic recombination. And that's before we even look at mutations. So in terms of natural selection and evolution, mutations are probably the most important cause of genetic variation because they introduce new alleles that have never been seen before. So not just new combinations of alleles, but completely new alleles. A mutation is a change in the sequence or quantity of nucleotides in the DNA. So the nucleotides could swap order, they could move around, or there could be one added, one deleted, multiple added, multiple deleted, etc. There's a lot of ways for mutation to occur. We covered these in previous videos. Mutations lead to a change in the amino acid sequence of the protein synthesized. So we looked at this during RNA, where we see that if the nucleotide of DNA changes, this will obviously change the nucleotide coded for in the mRNA, therefore change in the nucleotide in the codon and the anticodon, therefore possibly a change in the amino acid. And if there's a change in the amino acid, it could lead to a change in the protein. A mutation that occurs in a gamete or a gonad. So a gamete is like a sex cell, a gonad is a sex organ, so an organ that produces sex cells. These mutations will be inherited, they will be passed on to future generations, but a mutation in a somatic cell, so in a body cell, they're not passed on to the offspring. That means that the mutations we're concerned about in terms of evolution, obviously they need to be hereditary, they need to be passed on. So we're mostly concerned by gamete mutations. We call them germline mutations. There's two main types of mutations, which we did look at in a lot more detail previously: gene mutations and chromosome mutations. Gene mutations are a lot smaller mutations. They'll only occur within like 1 or 2 nucleotides, whereas chromosome mutations are much more big mutations. They usually have a greater effect. This could be the deletion of like a whole chromosome, an extra chromosome, or like an arm of a chromosome. So they're usually a lot more DNA that's affected. When we look at evolution, we classify gene mutations or all mutations into 3 categories. We mostly look at gene mutations simply because most chromosomal mutations, not only are they almost always harmful, but a lot of the time they result in a miscarriage. Obviously, we're not going to focus on that for evolution because miscarriages don't continue the population. So most mutations, especially for humans and animals, are harmful. If it's a lethal mutation, that's what we call a mutation that causes the individual to die. The change in the DNA sequence causes the formation of a shortened or a changed protein. So the protein is terminated prematurely, or it's changed in structure, and that means it's unable to perform its essential function. That's why it's harmful. We also have neutral mutations, also called silent mutations, where they have little to no effect on the genotype of the individual, or they may have an effect, but they may have a noticeable effect, but it doesn't impact how we survive. So that would be something like being able to roll your tongue or not. You can see it, it does have an impact, but it doesn't affect whether we're able to survive or not. So usually there's no visible phenotypic variation. Every now and then, as in the case of rolling your tongue, there is visible phenotypic, visible physical variation, but it doesn't affect the species. And then there's also the beneficial mutations. These are, of course, the ones we focus on for evolution because these are what drives evolution, drives natural selection. The DNA sequence and the function of the protein change, but this allows for new genes that increase the chance of survival in a specific environment. This is where natural selection occurs and these advantageous alleles are selected for. And then obviously there would be the individuals that have these new advantageous alleles are more likely to survive to sexual maturity, more likely to breed and pass on the genes, and therefore the amount or the proportion of individuals containing these advantageous alleles will increase over generations. When all of the organisms of the population have this new allele, we call the mutation fixed. That means that it's certain in the population. Almost all of them or all of them have it. Fixed mutations are the only gene mutations that play a role in evolution, because these are the only ones that we can see have been driven by natural selection and have actually had a certain effect on the population. Mutations lead to genotypic variations in the population, which may or may not appear in the phenotype. Examples of variation in population include the finches of the Galapagos Islands and the white lions. So obviously these are just two examples. You don't need to learn them off by heart, but learn the concepts behind them, so that if you ask a different example in the exam, you'll know how to answer it. So if we look at the finches, during Darwin's voyage, he observed 13 different finch species on the Galapagos Islands. Each of the finch species had a different type of beak. So we can see here that this has a very thin beak, this is a very thick, heavy beak, and some in between. They all have different food sources depending on their beak, and they all differ to the mainland Galapagos finches that were on the mainland South America around 1,000 kilometers away. All the finches on the mainland were seed eaters, and they will belong to one species. Their beaks were adapted to eating seeds. However, the finches on the islands, some of them may have been seed eaters. Other ones, as we can see here, were insect eaters, nectar eaters, etc. So they differed depending on the food sources of the islands. So the Galapagos Islands consist of many islands. This is a bit of background. It's not really that important to know, but basically what it's saying is that these are some of the theories for how the finches got to the islands. It possibly could have been that the current carried the ancestors of the finches. So the mainland finches, it carried some of the mainland finches on tree trunks or strong winds from the mainland to these new islands. When they reached the islands, there was virtually no competition on the islands because there were no other finches. There were possibly few birds, few other seed eaters or insect eaters, etc. Because these islands were very isolated. There were a lot of empty niches as in food sources available, and there was virtually no competition. That meant that new feeding opportunities were possible. So the finches didn't need to only eat seeds. There were also a variety of insects available, or flowers and nectar, and so on. As the finches multiplied, there were mutations in the population, and some of these finches with beneficial mutations were retained in the population due to natural selection. Obviously, we know that this takes many generations. It doesn't happen overnight. And we know that it's random. So the finches, it's not random as in the beneficial traits were selected for. That's non-random. But it's random as in the finch didn't wake up this morning and say, oh, look, there's an insect. Let me make my beak shape better for eating insects. It was just that one lucky finch got a mutation that had a beak shape better for eating insects, and therefore he was selected for. The finches with the beneficial mutations reproduced more successfully, and the favorable traits became more and more common and more frequent in the population. Mutations in individuals led to more variation in the population. The finches adapted to the new conditions, and gradually they developed different mutations, so different from finches on the other island and different from finches on the mainland. The different traits were considered beneficial in each unique environment. So, for example, one island might have had a whole lot of bees, and these finches adapted to be able to eat bees. This is beneficial on that island with the bees, but if the finches move to another island, which is full of seeds, this is not beneficial anymore. So it's only considered beneficial in the unique environment of each island. This genetic variation led to a variation in beak size. So the beak size is a phenotype, the physical characteristic that we can see, and that's one of the features that differed between populations. As more mutations occurred, each island's population developed into a distinct species that couldn't interbreed with any other species on different islands or with the species on the mainland. And we know from talking previously that this is called reproductive isolation. If we also look at the white lions, in lions, the brown fur color, so this fur color is dominant over the recessive white fur color. The gene for white fur color is a mutant gene, so it started from a mutation a while ago, and we know it's recessive and it suppresses color, so it suppresses this brown tawny coat. When two heterozygous brown lions, so capital B, smaller b, we know from looking at genetics that the trait exhibited in these heterozygous lions is of course brown, as this is the dominant trait. When they breed, there's a 25% chance that they'll produce a white cub, so it's homozygous recessive, carrying both white genes. This phenotypic variation, the phenotypic variation is of course the white color as opposed to the brown. It's not advantageous because these white lions are a lot, they struggle to camouflage. It's a lot more difficult for them to hunt. They're easily spotted by not only by their prey, but also by hunters. That would mostly be humans. Natural selection decreases the number of white lions because it's not an advantageous trait, and these white lions are unlikely to breed, not only because they are very unlikely to reach sexual maturity, but also possibly because lions may prefer to mate with tawny lions. I don't know if that's true, but it could be. So we know that the number of white lions in the wild is becoming more and more rare because it's not an advantageous trait. If we just lastly look at the founder effect, speciation can also be occurred by the founder effect. This is when there's a loss of genetic variation, when some individuals of a population move away from the original population, forming a new population from just a small number of individuals. And it usually occurs in a new area. So the founder effect occurs when a new colony is started by a few members, not very many, just a few members of the original population. The small population size means that the colony may have reduced genetic variation. So if we look at, for example, the ladybugs, there's less genetic variation because only the red ones have moved to the island. There's no variation in beetle color anymore. And it could be a non-random sample of the genes from the original population. So it could be, for example, we see here in the butterflies, there's brown, orange, and white butterflies. If we just took a completely random sample, we could have one white, one orange, one brown. But we see here that there's actually two brown and one orange. That could be just random, that 3 butterflies were picked up by the wind and blown to a new island. But it could be that the brown butterflies were the strongest and able to fly the furthest. So they were the only ones able to reach this new island. That's not random. So it affects the genotype and the gene pool of the new population, because now there's no white butterflies in the new population, so there's much less genetic variation. So founder effect can be good because in a very small population, if there's a beneficial mutation that occurs, it'll spread really quickly. But obviously it can be very bad because if there's a harmful mutation, it'll also spread very quickly, or if the environment changes and none of them are adapted, that makes it a lot difficult to be able to adapt. The gene pool of the new population only contains the genes and alleles of the founder members, so these few little members that moved to start this new colony. According to evolution, the isolated population may become so different to the parent population that it could form a new species. For example, if we look here in the dots, there's red dots and purple dots. A few dots of this old population migrate and colonize a new area, forming a new population. We call these the founder species or the founder individuals. Within this founder population, there could be a mutation. For example, this green mutation, which we say is favorable. Now this little green dot is more likely to survive to sexual maturity. It's got an advantageous trait, and it's more likely to pass on this trait. And eventually, over time, we have this new species that possibly could become all green. Now, the original population or the parent population doesn't have any green dots, whereas this new population has only green dots. Now they are unable to interbreed because they're new species. Some are the green species, some are the red and purple species, and they're reproductively isolated. So this can also be quite noticeable in humans, or immigrants move from one area to another and they remain isolated as a group due to something that could be religion, culture, language barriers. And this is where we could see the founder effect. For example, the Afrikaner population of South Africa, there was only about 40 Dutch settler families. They moved to South Africa in the 1600s. Obviously, we know this is a very, very small population, only 40 families, and they carried various genetic diseases like Huntington's chorea. They multiplied in numbers and they intermarried because they were so isolated from all the other groups that they only mated or married or interbred among their little group. So diseases like Huntington's spread really quickly. They also experienced a bottleneck, which is a drastic reduction in population, which further reduced the size of their gene pool. So now they have such a small gene pool, they have very little genetic variation. It was only a small sample of the original Dutch gene pool. So the modern Afrikaner population has quite a high frequency of the gene for Huntington's chorea and some other rare genetic conditions, which is a result of this founder effect from the original Dutch settlers. So if we just quickly compare the founder effect and the bottleneck effect, the founder effect just comes when there's an original population that moves away and forms a new population, whereas the bottleneck effect is when there's a reduction in size of the population because of environmental effects or human activities. So environmental effects, something like famine, earthquake, natural disasters; human activity, something like war or genocide. So the founder effect, usually the original population still remains, just in a separate geographic area from the new population. Whereas in the bottleneck effect, some new individuals remain, but the old population has almost always died out through something like this natural disaster or genocide or something. And we can see that the founder effect occurred in the Galapagos finches, where a few finches from the original population traveled to the Galapagos Island, survived, reproduced, adapted to the environment differently to the mainland finches. And we saw that there was this large population. Over time, there was this large population that resulted from just a few individuals, a very small gene pool from the mainland. Something like cheetahs could also be an example of the founder effect and also the bottleneck effect, where over time there was an ice age and almost all cheetahs became extinct, and there was also poaching, environment loss, which were both examples of bottleneck effects for the cheetah, drastically reducing their population. And there was also very few individuals remaining, possibly because they moved to new areas or broke off from the original population, which is the founder effect. And we now know that cheetahs have less than 1% genetic diversity. This is tiny. There's very, very little genetic diversity. So this is obviously not good for them because they're not able to adapt to the environment.